I'd like to start the drug delivery session. I'd first like to introduce Uday Kompella to come up and give a lecture here on the anterior segment side of drug delivery. And he'll be followed by Michael Goldstein on the posterior side, and then we'll have a panel discussion.
Okay, good morning, everyone. I would like to thank the organizers for inviting me. I'm at University of Colorado. I welcome you all to Denver. So on this slide, we have a number of topics that I've listed, but I'm going to focus on the blue bullets in the interest of time. So what's happening right now is a lot of innovations are taking place for long-acting drug delivery system. That'll be my focus. And I've listed some products that are already approved for the front of the eye, some that are in development, and we'll go through a few of these. This is a snapshot of the landscape of long-acting drug delivery systems, especially the implant-based delivery systems. You can see a lot of delivery systems are in place in the market for systemic therapies and some for ophthalmic conditions. And those that are being used for the eye are the smallest. And they also carry the lowest amounts of drug at about a few hundred micrograms or even less than that. And what you see on the left is one interesting systemically dosed delivery system that lasts up to 8 years. It's an intrauterine device, Minera, a contraceptive device. It uses about 52 milligrams of drug. So technically speaking, the concepts that we are talking about with long-acting delivery systems, specifically those based on membrane-controlled drug release, can be extended up to several years. However, the drug release will decline with time. Over 8 years, it declines by threefold. They're supposed to be zero order, but then they're not absolutely zero order. There'll be some decline over time. So next, we'll look at iDose. iDose TR is from Glaukos. We have already talked about it this morning. The device looks like this, and it's approved for clinical use. It's non-inferior to timolol up to 3 months. It can sustain the drug levels much longer than that. It can suppress the intraocular pressure for 12 months to the extent that patients do not need to take their drops as much. It's 8 out of 10 patients don't need the drops at 12 months. At 12 months, this device can be re-administered, which is an important advancement that was talked about this morning. The company is right now working on iDose TREX, which is extended iDose TR. So we hope that it will extend the delivery even longer than what we have seen so far. On the left, you have a graph based on this drug delivery system. On the right, what you see is the device placement between the cornea and the iris, and it is anchored into the sclera. So it's actually attached to the sclera, and it can be placed in and it can be replaced at the end of 12 months. So with respect to the drug delivery, what you see is a blue line which shows you the amount of drug that is remaining in the implant. This is a human study. It's very rare to see a human study for pharmacokinetics within the eye. So in this particular study, they have shown a gradual decline of the drug levels over 24 months at 3% per month. Decline is 3% per month. And also they're showing you aqueous humor drug levels on the right in red. And at the end of 24 months, the drug levels are still 1.5 times the Cmax that you will get with an eye drop. So what that means is you're maintaining significant drug levels at the end of 24 months with this device. With the new revisions, you can probably see even more interesting developments. And another system that was also talked about this morning has to do with the intraocular lens-based delivery system. I'll introduce that in a minute. But intraocular lenses have been used for drug delivery since 1985. And initially, they were soaked in the drug solution so that can sustain the drug delivery for a few weeks. And on the left, I'm showing you one device that was used in 2008 in preclinical studies, where they have attached 2 delivery systems to the haptic of the intraocular lens to sustain the delivery of triamcinolone acetonide. On the right, I'm showing you multiple ways you can attach the delivery system to the intraocular lens. The yellow circle, yellow rod, they all represent the delivery system that can be placed on top of the intraocular lens, leaving room for the vision axis. Basically, you can see through, but the device can be placed around the lens. So these kinds of technologies have been implemented over time. But what's interesting is, most recently, Spyglass has made some news with their 3-year bimatoprost release system, and that's what you're seeing in this slide. Here you can see two pads next to the intraocular lens. It is attached to the intraocular lens via the haptic, and it basically connects with the optic part. So it's basically latched onto the optic part. So these two pads are based on silicone. Most materials for these long-acting systems are based on silicone or ethyl vinyl acetate or polymers like that. So this one is based on silicone. And within this system, they have 75 micrograms of the drug, which lasts up to 3 years. Of course, you can imagine that drug-based side effects could be present. There is a surgical procedure done with cataract surgery, but something that I was able to think of with respect to some challenges are the weight of the pads, because the pads could add some weight to the device. How does that influence the settling of the implant or dislocation of the implant? That's something to keep in mind. And silicone as a material can stick to the capsular material. Would that lead to some alteration? That's something else to think about. Can you remove these pads once the delivery is done, or can you replenish the drug in the pads? So those are some of the questions that we have with these systems. And another one is here. This is based on a prodrug, a polymeric prodrug of latanoprost being developed by Polyactiva. And what they claim is that this particular prodrug, which is actually a new chemical entity, can be molded into implants, gels, and films. And I have taken one figure from their patent. And what you can see here is over 6 months, the drug is slowly released from the implant. It's latanoprost-free acid. Again, it can work for glaucoma. Glaucoma is a big market, so is dry eye. But right now, what I talked about so far is glaucoma-related new products that are interesting. So in conclusion, what we have seen is a number of drug-eluting implants are being evaluated. Some of them are very innovative. They may require surgical procedures, but still they're offering a great opportunity for extended delivery from 6 months to 3 years. 6 months ones are injectable ones. 3 years ones are based on surgery. And can we extend this up to 8 years? That's technically feasible. And then there are a number of questions that are still remaining. And one important question is, are these devices going to be better than drops in terms of patient outcomes for glaucoma? Thank you.
Thank you very much. Appreciate you.
Thank you.
Great. So I'd like to thank the organizers for allowing a cornea specialist to speak about the posterior drug delivery. What could possibly go wrong with this? That said, I have been a chief medical officer for a number of companies that have been developing in this space. Here are some of the companies I'm currently consulting with, both front of the eye and back of the eye. And it's really an honor to talk about a super exciting area and all the exciting things that are happening with delivery to the posterior segment. As this room is well aware, it's a huge multi-billion-dollar market. There are large opportunities like wet AMD, dry AMD, diabetes, and then there are orphan indications, a number of different orphan indications. The mainstay of treatment is intravitreal injections, with over 10 million intravitreal injections performed per year in the US, with over 3,000 ophthalmologists performing them. There are a number of challenges that we need to overcome. First among them is the blood-retinal barrier. We want to avoid systemic toxicity as much as we can. We want to target the cells, and that's a huge area of development here. And we want to relieve treatment burden. Patients don't like to come in frequently to get injections. So what are some of the goals across the board? We want to be able to target the right cells with the right amount of drug as infrequently as we can. To that end, we want better accuracy, looking for greater durability. We want improved efficiency, and we want to improve safety. And these goals will depend on what the indication is. So for example, in wet AMD, I would argue that the most important goal perhaps may be durability, whereas with inherited retinal diseases, we need better efficacy, and with dry AMD, I think we need both. So this is sort of a summary of all the different ways that people are approaching this problem. So and people are very familiar with this, any way around the eye. And then don't forget about systemic delivery mechanisms, and we'll talk about that in a moment. So the advantages of local delivery are well known. So as I said, the mainstay is intravitreal injection, which gives you high concentrations, in-office procedure, it's safe. That said, there are some risks, and patients don't love it, and there is the need to do redosing. Suprachoroidal delivery offers perhaps a better way to get to certain tissues in the retina, a little bit less well established, but still an in-office procedure. Subretinal delivery gives you really high local concentrations, but it requires a pretty complex surgical procedure, and there's sort of the risk-benefit trade-off. And then periocular and topical applications are still around, although the bioavailability is pretty poor. So what about systemic approaches? And I would argue, and people could argue against that, for a long time we spent a long time looking at systemic drugs and saying, how can we deliver them locally? And then that was sort of an approach that a lot of companies took. And now it's a little bit back to that. Now people are looking back and saying, well, what about systemic delivery? Maybe that would be a better approach. The advantages really are that you can treat both eyes at a time, and there are new ways of delivering drugs systemically that minimize systemic adverse events. This is particularly happening in dry AMD and some of the visual cycle modulators. So we also have all sorts of different treatment modalities. All of these are active in the posterior segment world. With examples here, small molecules give you, are well known. They're easy to manufacture. The issue is that you need to dose frequently, and there's the risk of off-target effects. Peptides and biologics, much more specific, much more targeted. However, you have issues with manufacturing. They're more expensive, often cold chain. You get immunogenicity issues. So those are being worked out. And then sort of the newest ones, I think, in ophthalmology have been cell therapy and gene therapy. Those really offer the opportunity for disease modulation, and actually you can actually have cures. They can be a little bit complex to deliver, and again, the issues really around the manufacturing issues are fairly complex. So here's an example of one of the problems, so solving the extended drug delivery problem, and there are sort of three main approaches that people are using. So the non-bioerodible implants, there's approved products, there's products in development. Advantages, they're easy to put in. You can get sustained delivery for a long period of time, but the disadvantages are you have a foreign body. Biodegradable implants, again, there's approved products and several that are in late stage development. Advantage, again, being that they're easy to put in. Disadvantage being that you actually have to do repeat dosing. And then there are several companies working on a no implant design where you take the drug, you attach it to a scaffold, and then the drug will release over a period of time. So, key takeaways, this is a very large, exciting market. Currently, local delivery does dominate, but I think there is a bit of a trend back toward systemic delivery. I think durability is sort of the next frontier, and there's a number of companies that we're going to hear about today throughout the course of the day working on this. I think the most exciting thing is we have a number of diverse modalities and delivery methods, and so depending on what type of indication we're seeking, we can actually come up with the best sort of drug products developed for that. So thank you.
My co-moderator, Ross, to introduce himself, and then we'll come down the panel introducing themselves, and then we'll start the discussion.
Thanks, Bob. I'm Ross Ethier, faculty member at Emory University in ophthalmology. Jay?
Hi, I'm Jay Duker, President and CEO of EyePoint.
Hi, Austin Blackwell. I'm the U.S. Head of Marketing for Retina at AbbVie.
I'm Michael O'Rourke, CEO of Re-Vana Therapeutics, based in Tampa and in Belfast, Northern Ireland.
Hello, Rich Small, I'm CEO of Neurotech Pharmaceuticals.
Tomas, Navratil, Chief Development Officer, Glaukos.
Thanks, and Chris Brittain. So I head up clinical development for ophthalmology at Genentech and Roche.
Thank you, everyone. I'd like to start by asking Jay a question about what is the general overall view of the benefit of sustained drug delivery, in your opinion? I mean, is it just less treatment burden, fewer visits, or is it better outcomes, or is it both? And the second part of it is sometimes there's a trade-off in that outcome with more of a side effect profile or inflammation or something that you give up to have the sustained delivery. So how do you play that factor into your evaluation?
Do I get all the remaining 30 minutes to answer that?
No, you get 30 seconds.
Great questions. And I want to say, and I probably will speak for all the sustained release companies, I think we now believe that we will get better outcomes in the long term. I mean, there's been data for years that we're all aware of that in the real world, patients lose vision, they don't do as well as clinical trials, and the more injections you give, the better results you get. I think there was some worry that if you suppressed VEGF long-term constantly, there may be adverse events, but I don't believe we've seen any strong clinical evidence of that. So I think that the paradigm is shifting, that it's not just fewer injections, it's actually better outcomes in the long term by not allowing the VEGF to do the leakage issues that we know it will over the long term. I think the added benefit is not necessarily fewer injections. I think it allows the doctors and the patients to modify the schedule, should they choose to one that makes more sense.
Thanks, Jay. Chris, can you comment on that from your perspective? Because you have this great sustained delivery situation, which may have better outcomes with the PDS, and but you had a little bit of a side effect hurdle to overcome. Any comments?
Sure. No, I think sustained delivery or continuous release, however you want to describe it, and the different mechanisms for different platforms, which I think is important. I think it's both great for the patient and for the healthcare system. So I'll give you two concrete examples. If you remember the 7-UP study, where we see 7 years of data from IVT injections, patients lost 20 letters, 4 lines of vision from their best visual acuity outcome. Susvimo, which is continuous release from their best visual outcome, 7 years later, they lost between 2 and 7 letters, so it's a significant improvement. Patients are still losing vision, so there's still a massive unmet need in NAND. And then to your question really about the safety profile, and I think this is where we really focus on partnering with physicians, understanding how the technology is used. So early on in the Susvimo development, we saw this as the implant was put into the eye, we saw like a 50% vitreous hemorrhage rate. Then we stopped the program, we went back to the drawing board, did some additional surgical optimization with some experts, and that vitreous hemorrhage rate went down to 5% in that phase 2. Now it's even lower with even better technology. So partnerships, understanding, and really, I think you've heard on the previous panel, transparency about the safety and what patients and physicians can expect.
Thank you. Austin, with respect to your company's permonidine implant, the initial one where you could use it but only once because of safety concerns, do you have any insight into, how to find these problems earlier and how you have adapted and tried to make something that's much more long-acting, which is under development now?
Yeah, I think that a couple things. One is just really paying attention to what happens in the earlier trials, phase 2, and using those learnings to inform, our different sort of approaches for phase 3. I think the other thing is really looking and using something like IRIS, the IRIS database, to really, pull the amount of patients that are in there and use them to, an advantage to inform future trial design.
Maybe that's a natural segue into a broader question around that, which obviously there's a lot of exciting technology in this space. We've heard about some of that already this morning, and the question is, how are these novel technologies changing your clinical trial design and your regulatory approach? Obviously, having implants that are in there for a long time, there's complications or more complicated surgeries, potential for inflammation, migration, atrophy, etc. Tomas, maybe I could ask, from the Glaukos perspective, obviously you have some experience in this now. I'd love to hear your comments.
Thank you so much. So I'll use 2 examples. Our iDose TR intracameral implant that has demonstrated 3 years of IOP lowering after a single dose. And maybe second example is our intravitreal retina implant that targets wet AMD and DME patients that showed 4 years of vascular leakage suppression after a single dose in preclinical models. So drug delivery and extended drug delivery is bread and butter what we do at Glaukos. And of course, we optimize each clinical trial design and regulatory strategy uniquely to that extended release product. So maybe there are 4 specific examples. From the standpoint of efficacy of our products often exceeds the requirement for primary efficacy assessment by the US FDA. So what we often do is we declare secondary key efficacy endpoint and keep the study masked until we reach that key secondary endpoint, which succeeds, let's say, the 3-month primary efficacy FDA requires for glaucoma studies. Second is the re-administration, which is really critical. We built it in from the start into our pivotal studies, and that enables us to file for re-administration at the point of original NDA submission. And so that's really important. And FDA recently indicated iDose TR is the only and first-in-class intracameral implant that has such label and allows re-administration. Maybe third one is utilizing PK that Uday very nicely highlighted in his presentation. We collect PK in the course of pivotal studies as well when we re-administer the implants, and so that enables us to analyze the amount of active in the aqueous humor and to reassess how much payload is left. And lastly, we keenly focus on safety because the therapy is not a whole without providing sufficient safety and studying enough patients long enough to fully understand the safety profile. So these are 4 examples.
Great, thanks. Rich, anything you'd like to add to this? And then Michael?
Yeah, I mean, so what we do is we actually do a little different variation, and we actually encapsulate cells into a capsule that's designed to actually release the factors over an extended period of time, while also allowing the nutrients in the eye to continuously produce those. So we've actually have had, because we've actually have done programs in other diseases, so we have quite a bit of history on these implants, and we've actually, the longest dated implant that we've explanted was 14.5 years, and we were still showing viable cells, and they were still producing at basically the same level as it would have been at the day it was introduced into the eye. Our safety profile has been very well tolerated, and we still have patients, believe it or not, that still have implants in their eye for now, probably over 20 years. And so we don't have the same concerns as far as what we're facing with safety rejection. We have not seen that. And in our clinical studies that we got approval for Inselto, during the study, which ran 2 years, there was not one single implant that got explanted over that period of time.
Michael?
Yeah, so I've been in the ophthalmic drug delivery space now for 30 years, originally launching Vitrasert back in Chiron days, 1995. And really then, there was, for CMV retinitis, the key thing we were looking at then was really inflammation, but with an antiviral drug that was suppressed in many ways. Another 9 years later, we launched a Retisert, which was a Bausch & Lomb, but did have a steroid in it. So the biggest concern again was inflammation. Both of those were implantable, surgically placed implants. So there was a lot of cases of cataract formation, IOP that led to certain trabeculectomy cases. So those were safety issues, but were generally managed. Really with the progression into clinic-driven drug delivery implants. I was at Greybug when we worked with the microparticle, nanoparticle technologies with Sutan. Wasn't so much an inflammatory problem, there were some visual acuity problems because of some of the particles getting into the visual axis. And now fast forward, really with Re-Vana, we were looking at sustained release biologics, small molecules, complement drugs. One of the most important things we look at, and it's actually the biggest killer of drug delivery technologies, is poor tolerability. If you have inflammation in the eye, it really does kill the technology. So for us, tolerability is really one of the most important criteria, beyond other safety factors when you're dealing with biodegradable implants.
Thanks,
I have a question maybe more on the retina side here. We have a lot of things to choose between right now, and hopefully we'll have a lot more in the near future. But Jay, when you're trying to pick the most clinically relevant endpoints for a trial, they don't always sort of show us what to adopt in the real world. Sometimes those endpoints aren't really representative of what we really want to know when we're choosing to use a drug in the clinic. They may be more geared towards getting approval only. Do you think there's a way of picking endpoints that would be really clinically relevant for us to decide which agent to use, or is it more a game of just getting approval?
The short answer is you need to get approval or you're going nowhere. And so the agency has been very clear with posterior segment drugs, with just a couple exceptions, that it's a functional endpoint. It's a visual acuity or visual field. It's not structural. And yet the retina community, we make treatment decisions largely based on OCT, which is structural changes. The linking of the two, the agency for years has said, show us data that says OCT is going to be a good predictor of visual acuity and we'll accept it. So I think from a drug development perspective, you have to get the drug approved, but you have to recognize that all of this goes into a clinician's decision is, am I going to use the drug and in whom? We talked a little bit about safety, but of course you have to look at the disease you're treating. If you have an inherited retinal dystrophy that's going to cause blindness in a child, you're willing to accept a certain degree of safety issues versus wet AMD, where we've got some very good, short-acting, but very good, safe drugs. So it's not a one-size-fits-all, and I think that both clinicians and patients are willing to have a set-off for more longevity and the potential for longer-term visual acuity if there might be some safety issues that are a little bit higher in the new drug than there was perhaps in the old biologics. But that's all a decision that gets made after approval. So I go back, you're bringing up a great point, but the practical aspect from drug development right now is you gotta listen to the regulatory agency.
You get it approved and we'll figure out how to use it, I guess.
As long as we get paid.
Okay, just following on that, how about in the anterior segment? So, you know, it's a slightly different world. Anybody want to comment on that? Thomas, you have a lot of experience there, but maybe opening that up a little bit as well.
So in the anterior segment in glaucoma, we have a very straightforward path. FDA has very early established, and CEDAR has been very consistent and clear over the last 30 years, what they require for primary efficacy. For IOP-lowering agents. I think for neuroprotection, it gets a little bit more complex and exciting. We have a very high bar from US FDA regarding improvement in visual fields. I think there's ongoing dialogue whether there would be kind of an additional endpoint that they would recognize. But the bar is very clear for IOP-lowering efficacy over the first 3 months, usually at day 15 or day 14, week 6 and week 12. At least twice of the day to capture the diurnal cycle. But for neuroprotection, we feel there are more challenges, and hopefully a couple of these early trailblazing programs will establish the change in visual field, but also additional changes like changes in slope could be acceptable as a primary efficacy outcome. And neuroprotection is where the future of treatment of glaucoma is.
Yeah, I think the number one barrier is regulatory. Lots of great ideas out there, but getting it to market is challenging. Any other comments in that area? Okay, let me change gears a little bit and look more at the whole pipeline here. And I'd actually like to maybe kick it off with Austin here, and then hear from other folks as well. From your viewpoint, what do you regard as the rate-limiting step getting the new drugs into the clinic? Are you limited by good basic science? Is it, more capital needs, more and more complex, longer, more expensive clinical trials, regulatory complexity, just too much as we move into some of these new technologies? I think I can guess that probably you're going to say it's all of them, but I'd love to hear folks' perspective on kind of what's top of mind in that list.
Yeah, I think you're right. It's all of them. One of the things that really comes top of mind just hearing that question is how to operationalize something that's new and novel into the clinic setting, right? I agree with Jay totally. You've got to get approval. That has to be the by and large goal. But if it's a different delivery mechanism, what is that going to look like in the day-to-day? And it may look different than the current standard of care. So how do you kind of do the work in advance to make sure that you're carving out what the new normal can look like, and make sure that it works for all stakeholders, so that you're sort of enhancing the current ecosystem to what it could look like in the future.
It's good to have a crystal ball. Chris?
No, thank you. I think I'm going to slightly disagree. I don't think the regulatory approval is the end. I'm not sure if that's what you're saying, but for those of you who remember ochraplasmin, we got that approved. I worked on that program for kind of 18 months back in the UK, and that was pretty much a disaster for us at Alcon and for patients because of the safety, and there was unclear safety data there. So I think we can get therapeutics approved, and then the safety isn't good enough. So I think for me, a lot of what we work on is really de-risking safety early in a trial program. So I think Michael talked about it. If you see signs of inflammation early in a program, you really want to know so you can quickly kill and redevelop and relook at the biology or re-understand what's going on. So I think number one for me is really understanding the safety. And then on top of that, I think you heard in the last podium panel, or the two panels before, and you'll hear from the FDA again this afternoon, which is these opportunities to reduce the white space with these quite exciting pilot programs. I don't believe what Marty Makari said, which was we can reduce it by the FDA alone by 50%, because there's not that much white space in drug development. But I think there's significant opportunity there to really narrow down some of these timelines through faster discussions and faster decisions. And then the last thing I'd say is 15-letter superiority or 15-letter losers versus gainers is fine in the US. It's not fine necessarily elsewhere in the world, Europe, Australia, China.
Rich, and then maybe Rich, and then Michael. Rich, kind of from the cell-based perspective?
Yeah, so let me take it from a little different perspective, because obviously Neurotech is a much smaller entity, and we're in the middle of we're considering a very successful launch, but we're less than a year into it. So really for us, it's about focus rather than just spreading out as wide as we can go, because we don't want to really misstep on the launch itself, and so that's a really big factor for us. We know, because we have looked at our technology in other diseases, and almost anything that you want to deliver long-term biologics into the retina, we can actually have a solution for. And we actually have data for other programs too that we're very interested in. But for us, it's more around just focus and sequencing rather than just some of the other aspects that maybe larger companies would think about. So that's more the way we look at newer development or developing newer programs.
Great. Michael?
Yeah, so it really is, to answer you, is all of those things, but I think it also depends on what stage the company's at. Re-Vana is an academic spin-out back in 2017. So we really started from complete scratch, almost from like a PhD thesis. So the challenges beyond all the issues you mentioned are clearly to raise capital, is to get the company funded. And that one really comes with proof of concept. So you have to balance the capital you raise with how you're demonstrating progress of your technology. In our case, we were also taking on something novel. We're looking at the sustained release of biologics using photo-cross-linking with light instead of heat to power the implants, but these implants to be delivered in the clinic, so without any surgery. So you put all those together, and you're at the very early stages of a company and how you then build it. But we do have really good investors behind us now. We raised about 16 million. Some are here today, Vision Ventures, Excite. So that really, in our case, was beyond all the other challenges. It's really getting a company started from scratch.
And that's a great segue into the financial side, I think.
Well, it's, developing these agents, these sustained delivery or slow release agents is expensive, in part because of the duration of the trials. These trials, can be extremely time consuming if you have a long-acting agent that's repeatable, and the FDA wants a long trial. They're expensive to do, and we have a lot of startups and entrepreneurs out there. Other than raising the typical venture capital, one option is strategic partnership, and the question is, when to do that. And I'd just sort of like to hear Michael, your experience from your recent strategic partnership and how that's working out for you as another alternative to just raising typical VC funds, which have been tight the last few years.
Yeah, thanks, Bob. So, I mean, with the companies I mentioned, from Chiron right through to Re-Vana today, partnerships has always been extremely important. Even when we launched Vitracert back in 1995, we had a partnership with Roche to get the drug, the antiviral drug, Genentech. We controlled delivery systems from Boston, who gave us the technology. So partnerships are really important from day one. But if you fast forward now to Re-Vana, so in July 2025, we did sign the strategic collaboration, a licensing deal with Boehringer Ingelheim. Members of the team are here today. So Boehringer is, one of the oldest pharma companies from 1885. And we knew Boehringer were building up their Retina pipeline. So we gave this talk actually at the OIS last year. So from the first introduction really to signing the agreement took almost exactly 2 years. So the deal here is, we have Re-Vana's two different technologies. One is iLeaf, one is an in-situ forming implant, Oculeaf, which can deliver 62.5 milligrams of drug without really compromising the needle size. So we can get a much higher payload, 50 times greater than some other therapeutics. So the job we had then was to work with Boehringer and to agree a deal whereby we end up with up to 3 targets per year from the Boehringer portfolio, and we look to see if we can match those against our technology, either iLeaf or Oculeaf technology. But this is really all milestone based. But so far, the relationship, to your question, Bob, has been really fantastic. Incredible company to work with, Boehringer Ingelheim. I think we have a mutual understanding as to what we want to drive to, what we want to get to. And ultimately it is reducing the treatment burden. But we're also looking beyond just stainless biologics, either established molecules. We're also looking at novel drugs as well. The relationship is target specific. So we can work with other collaborators as well, if need be. But today, the relationship with Boehringer has been transformational for Re-Vana. We said that in a press release, and they've been a really fantastic company to work with.
Do any of the other panelists have recommendations for startups as to when to try to seek a strategic partnership, a build to buy or anything like that? Any suggestions from the panelists? I
don't know that I have a suggestion except to say talk to a lot of people and a lot of entities, because it isn't a one size fits all. And the macroeconomic environment can change overnight. And equity financing may be available one day and next week it's not. It's also very much data driven in that I think you need to think about planning a phase 1 trial, unfortunately not just to show safety, but you have to have some hint of efficacy or you're not going to be able to fund the phase 2. Strategic partnerships can be important at any stage. And again, I think one should keep an open mind. But at EyePoint, we've been able to raise over $600 million in equity over the last equity financing over the last 3 years, and we've been a commercial company before, and we're gearing up to do it again. And so for us, it didn't make sense to have an early partnership, but it certainly can for others.
Maybe for Glaukos as a strategic, we are stage agnostic, so we are looking at the preclinical stages, clinical stage companies, as well as commercial stage, and we have deals in each segment over the last couple of years. But I think what is important is that the foundational therapy and foundational technology truly delivers. And so we often encounter teams that have 10 patient study and they are ready to go to phase 3, and we often find that there's less progress than we would expect based on the original messaging. So I think an advice for the early startup companies, just have realistic expectations how the risk your technology is. That's an important piece of the process and the important process of the dialogue.
If I may, just 2 or 3 points. Number one, please talk to us early. We want to look at all of these different technologies. I think that's really critical. And we've, specifically Genentech, we've got a lot of scientists who've worked on these delivery technologies with many of you in the audience over many, many years. 20 years we've been trying to have these long-acting delivery technologies, and we've got quite a lot of understanding. They are really hard to develop. So, naked LED technologies in animal eyes to de-risk safety is critical. People tend not to do that. So I know it doesn't get you money necessarily from the startups, but in the longer term, it will get you much more interest. And then you can start sticking your assets onto those. And, you know, we're interested, and I think this is probably the case for all strategics, is your platform good for small molecules, for intracellular delivery? Is it good for just long-acting retention studies within the vitreous, or is it for the front of the eye? So I think you've got to come to us with, you know, what's your aim, but where else can it be used, and how have you de-risked it?
Okay, we need to be careful here not to transgress the Gil's timeline. So going to be one last question, and I'm going to ask just for real quick responses on this. So, you know, we've heard about some interesting technologies. Michael, in his talk at the beginning, talked about suprachoroidal. We haven't talked about punctal plugs for the anterior segment, et cetera. Anybody got any, let's call it non-traditional routes that excites them that they're thinking about? We're just going to go down the line. Jay, anything good?
I think everybody on the panel, one could label much of what we're doing as non-traditional. I think that all of these potential sustained release may play a role. And so I think the other things to think about are not just delivery of what we have had already, but do MOAs. Anti-VEGF has been great, and we've had it for almost 20 years. But we've always believed that there are different MOAs out there, and in that I think it's not just drug delivery, it's the ability to block other receptors or other pathways that are going to get us further forward.
Austin, 20 seconds, sorry.
Yeah, I think that they're all exciting. All the routes of administration are exciting. And I think the concept of the continuous delivery to, you know, sort of disrupt and avoid the, you know, some of the challenges with the bolus injections and peaks and troughs is very exciting, both from a outcomes perspective with vision, but also from an anatomical standpoint.
Great. Michael?
Sure, I mean, for us, it's still intravitreal, but with our Oculeaf technology, it's really, we're injecting a liquid into the eye and photocross-linking that with visible light, we form an implant with a special applicator that we've developed. So as I say, with we can get a huge payload, it's 62.5 milligrams of drug with a 25 gauge needle. We think that is going to be really groundbreaking as we move forward in drug delivery.
Rich?
Personally, I would just like to make sure that we leverage our encapsulated technology into other disease areas.
Thomas?
We are excited over many non-validated ocular drug delivery technologies, but the proof is in the pudding, and they are yet to be validated. What I'm excited about is development of new chemical entities specifically targeted for extended drug delivery, new polymers that enable us better control of the drug delivery, and novel particle engineering that overall enables us to make more safe and effective product for our patients.
Chris, last word.
I've run out of time. Just one thing then, bispecifics, trispecifics, combining novel biologies to have much more durable outcomes, I think is exciting.
Okay. I'm going to have to save this for the next Eyecelerator. On behalf of my co-chair, Bob, I want to thank all of our panelists and our introductory speakers. Let's give them a round of applause.
Thank you, guys.
Thanks, everybody. And we're going to move on to our next panel now. If they could please come up to the stage. Thank you.